Hello everyone, this is Raju signing in, and with that, from the world of life sciences, I present before you another exciting topic, that is DNA replication taking place in the eukaryotes. In our last venture of DNA replication process, we have analyzed the DNA replication process taking place in the prokaryotes, and now it's high time we take a step ahead by understanding and analyzing the DNA replication process taking place in the eukaryotes. And just like the other journeys, we are also going to dig deep into this very journey of understanding the DNA replication process in the eukaryotes. Now, since the entire DNA replication process cannot be adjusted into a single video, therefore, I have decided to segment the video into certain modules and submodules. Here, in this very module, we would be analyzing the very prominent structures of the DNA replication process, starting with the origin of replication and the different types of DNA polymerases that do play a very prominent role in the same. Before moving ahead with the details, let me give you certain glimpses of what exactly we are going to do in this particular video session. Now, we very well know that the RIC is the origin of replication for the prokaryotes, but in case of eukaryotes, it's a bit different. And therefore, it is not known as RIC, it is known as autonomously replicating sequences, that is ARS. Now, within the ARS, there are certain domains and subdomains, and also we are going to understand that what are the exact locations and the importance of these domains and subdomains, and how it does play a very prominent role in initiating the very miraculous process, that is the replicatory process in the eukaryotic DNA. Not only that, we are also going to analyze the different functionalities of different types of DNA polymerases playing a role in the same. Now, the DNA polymerases which we are going to discuss in this video would be of two different types. In the first section, we are going to discuss about those DNA polymerases that do play a very prominent role in the nuclear replication process. And the DNA polymerase which we are going to discuss at the next is the DNA polymerase which do play a very prominent role during the replication process taking place in the cellular organelles such as mitochondria and the chloroplast. So, without any other further delay, let's begin the most exciting journey of understanding the DNA replication process taking place in the eukaryotes. Hope you would love it. So, let's start our journey of understanding the nuclear DNA replication in the eukaryotes. And starting with it, now we need to understand that the nuclear DNA is linear in nature, which supercoils itself into a structure which is known as the chromosome. Now, the replication starts from the origin of replication, which in case of eukaryotes is known as the ARS, or the autonomously replicating sequences. And this replication starts during the G1 phase of the cell cycle. Now, in order to understand the replication, that too nuclear DNA replication in eukaryotes, we must consider a representative so that our understanding about the replication process becomes very concrete and well assembled. So, a representative is considered for the eukaryotes, and it is one of the simplest eukaryote known as the buddingest. Now, while understanding the ARS, which is the origin of replication for the eukaryotes, certain points needs to be very well cleared. First point, that the eukaryotic origin of replication known as the ARS is shorter than the prokaryotic origin of replication, that is from the RIC. Now, if you ask me how much, then I would say that if the prokaryotic RIC is approximately 245 base pairs, then the eukaryotic ARS will be approximately 200 base pairs. So in a way, the eukaryotic origin of replication is shorter than the prokaryotic origin of replication. Now, unlike the prokaryotic origin of replication, eukaryotic origin of replication, or the ARS, has approximately 400 ARSs in the yeast genome. But In case of prokaryotes, we have seen that there was only one RIC. But unlike that, we have got approximately 400 origin of replication in the yeast genome. In other words, the eukaryotic genome is multi-repliconic in nature. Multi-repliconic in nature. That means it do possess more than one independently replicating origin of replication known as ARS. So, having said so much about the ARS, let us understand about the structure of the ARS. Now, this is a very schematic representation of the double-stranded structure of DNA that do possess the ARS. Now, looking into its diagrammatic representation, we can see that there are certain domains. Now, these domains are namely A domain, B domain, and C domain. Now, we have also seen that this B domain is further divided or classified into certain subdomains, namely B1, B2, B3. So, having understood about the very basic structure of the ARS, that it contains three basic domains, though it shows diversification in different genomes, but it does share a very basic structure, and this basic structure contains three domains, namely the A domain, the B domain, and the C domain, where the B domain is further subclassified into B1 domain, B2 domain, and B3 domain. Now let's look about the functionalities of these different domains present within the ARS. Talking about the A domain, basically this particular domain contains a consensus sequence which is basically AT rich, and this consensus sequence is known as the ACS. Now if you are wondering what does ACS stands for, then I would love to say that ACS stands for autonomously replicating sequences consensus sequence. Now basically this consensus sequence, which A domain is 11 base pair containing AT rich region. Talking about the B domain, it contains DNA unwinding sequences, which makes it very obvious that the initiation of DNA replication will start from this B domain, that is the melting process, which is very well required for the DNA replication process, will start from the B domain. And finally, coming to the C domain, it is present in certain ARSs. That means not all the ARSs possess this C domain. Now talking about the function of this C domain, then I would like to say that this C domain actually provides a platform for certain DNA binding proteins that may help or that may aid in the process of DNA replication initiation. That is, it acts as a stimulatory factor. Now, having understood the very basic structure of the ARS, let us now understand and analyze at the same time that how exactly the DNA replication process gets initiated from this very ARS. Now, talking about this, then at the first and foremost stage, there is a protein complex which is known as the origin recognition complex, which is basically a complex of 6 polypeptides binds collectively with A domain and B1 domain. With the binding of the ORC, there is a binding of another protein known as the CDC6 protein. Now, this protein binds during the G1 phase of the cell cycle, and during that phase it is bounded to ATP. Now, this protein, which is known as the CDC6 protein, while being binded with ATP, it interacts with this ORC complex. Now, this particular CDC protein have ATPase activity, which do means that it can hydrolyze its bound ATP and utilize the energy obtained from it from some or the other purposes, and as such it helps in recruiting the MCMs. MCMs stands for mini chromosome maintenance. Now you might be wondering that what the hell are these MCMs? These MCMs in case of eukaryotes will act as a helicase, and it is this CDC6 protein which help this MCM bind to this complex which is known as the origin recognition complex. Now this complex, that is ORC, is now complexed with CDC6 and MCMs. Now MCMs being the helicase will have to bind to the DNA template strand in order to perform its activity, and for that we need a helicase loader, and for that it is these proteins, that is CDC6, ORC, and another protein which is known as CDT protein, which will help loading this MCM from the 3' to 5' direction of the leading template strand, thereby helping this MCM to perform its melting activity from this particular very region. So since we have already understood about the very basic structure of the origin of replication for the eukaryotes, that is the ARS, their different domains and subdomains, their functions and so on, now it's time to understand the different types of DNA polymerases that do play a very prominent role in the DNA replication process of the eukaryotes. Now, in our initial lectures, it was already stated that approximately more than 10 different types of DNA polymerases are found in the eukaryotes. But here we are going to discuss about those DNA polymerases which has got a very prominent role. Now, starting with the discussion, I would like to say that the DNA polymerase which do play a very prominent role can be classified into two different classes. In the first class, we would keep all those DNA polymerases which do play a very active role during the nuclear DNA replication, and on the other class we will like to keep all the DNA polymerases that do play a prominent role during the extranuclear DNA replication, that is in mitochondrial DNA replication. So starting with the DNA polymerases of nuclear DNA replication, we have got DNA polymerase alpha, DNA polymerase delta, and DNA polymerase epsilon. Talking about this DNA polymerase alpha, structurally it is a heterotetramer. Now this DNA polymerase alpha is a very unusual kind of DNA polymerase, as it does possess polymerase as well as primase activity. Now with the help of primase activity, it can synthesize the primer, which is approximately 8 to 12 nucleotides long, also known as the initiator RNA, and with the help of polymerase activity, it can help in the synthesis of initial few nucleotides and add those nucleotides to the newly synthesized stretch of DNA, and that very short stretch of DNA can be approximately of about 20 to 40 nucleotides, and it is known as the initiator DNA. Now remember that this DNA polymerase lacks the exonuclease activity, which is very much important for the proofreading action, and thereby this DNA polymerase alpha lacks the proofreading activity, and hence there is no proofreading activity for this DNA polymerase alpha. Now having said that, after the synthesis of this initial DNA sequences, now it cannot further extend the newly being synthesized DNA, and as such it requires another DNA polymerase to join in, and thereby DNA polymerase delta and DNA polymerase epsilon, it joins in, and thereby DNA polymerase delta comes in. Now this process where the DNA polymerase delta comes in place of DNA polymerase alpha is known as polymerase switching activity. Now as the DNA polymerase delta joins in, it can now continue the elongation process of the newly synthesized DNA. But as already stated earlier in my previous lectures, the DNA polymerase has got a tendency to come off, to get detached from the template strand. Therefore, it is very much essential that it associates some proteins which will act as a clamp for the same DNA polymerase. Therefore, it recruits a protein known as pCNA, which is known as pCNA. This DNA polymerase delta recruits a protein known as pCNA, that is proliferating cell nuclear antigen, and this acts as the sliding clamp. It acts as the sliding clamp for this DNA polymerase delta, so that this DNA polymerase delta doesn't get detached from its template strand and thereby help in the higher processivity rate of this DNA polymerase delta. And for the loading of this sliding clamp, we require a clamp loader, and that particular clamp loader is known as the replication factor C. The clamp loader is known as a replication factor C. Now it is this particular clamp loader which will help loading this sliding clamp associated with the DNA polymerase delta to the template strand, thereby helping in the higher processivity rate of this DNA polymerase delta. Now, structurally speaking, this DNA polymerase delta is a heterotetramer, and it has got both polymerase and exonuclease activity. That is, this particular DNA polymerase delta, unlike the DNA polymerase alpha, has got the proofreading activity along with polymerization activity. Now, similar role is played by the DNA polymerase epsilon. Now, having understood this, let us also understand the second class of DNA polymerases which do play a pivotal role in the extranuclear DNA replication, that is in the mitochondrial DNA replication. Now, the enzyme or the DNA polymerase which do play a pivotal role during the extranuclear DNA replication is known as DNA polymerase gamma. Now, this DNA polymerase gamma is heterotrimeric in nature. So this is the difference between the DNA polymerase gamma and the rest of the DNA polymerases which are present in the eukaryotes. That is, the rest of the DNA polymerases are heterotrimeric in nature, but this DNA polymerase is heterotrimeric in nature. So having understood about the DNA polymerases which do play a very pivotal role in the eukaryotic DNA replication process, in our next video we will start the DNA replication, that is the initiation process in details. And till then, stay tuned, and of course, if you have loved the content in this video, don't forget to like, share and subscribe. So with this, I would like to wrap up today's session. Thank you very much for your patience.